Good morning, friends. Very good morning and welcome to Unacademy. This is Abhijit, and today we are going to discuss about the plant growth. Okay, so guys, are you there? So let's start. This is Crack Maharashtra CET channel, guys. If you do not have subscribed, first of all, subscribe the channel and click on a bell icon. So you will have a notification prior to the lecture, and with the notification you can attend the live lecture. So try to attend the maximum live lectures so you can if you interact with the educator, so definitely the level of understanding will increases. Okay, before starting the lecture it is necessary to know about me. This is Abhijit Madkar. I have more than 6 years of experience in the field of biology and also crack net CSIR with all India rank 60 from life science. Guys, we are here on the India's largest platform. If you do not have this app, first of all, install this app in your mobile. This app will give you the thousands of different uh, lectures from the thousands of different educators. So if you find your, may you find your favorite educator here on this app, and you can attend the lecture of that particular educator. Okay, या ॲपची लिंक तुम्हाला खाली डिस्क्रिप्शन मध्ये दिलेली आहे मित्रांनो. ओके, सो द लिंक इज गिवन बिलो इन द डिस्क्रिप्शन ऑफ दिस व्हिडिओ. So you can find the link and you can download this app. App is available for both, I mean the iOS and the Android also. Okay, this is Telegram app, guys. This app will give you the notes and MCQs and the links. So if you have a Telegram app already, so just join this channel Crack Maharashtra CET, and the URL of this channel is here and also given below in the description. So you just want to click on the very good morning beta. Okay, RR. A link of this channel is given below in the description. Okay, so guys, this is the final. This subscription will give you the the maximum use of this app. So I've already told you about the app of Unacademy. This app, you can use this app unlimited with the subscription. If you see the subscription of 12 month, the fee is 14,850 rupees, and the subscription of 24 month fee is 21,600 rupees. So guys, if you see as per the fee, so this 24-month subscription is more convenient because the fee is very in 700 to 800 rupees. So you are just paying 700 to 800 rupees per month. Okay, so guys, if you use this capital Abhijit as a subscription code, it will again reduce your fee by 10%. Okay, so here for 24 month it's a 14,850 rupees. So with 10% discount you will have the 1,485 rupees discount on it. This, so you are just paying about 1,300 rupees for a 12 month. Okay, so when you are taking a subscription, try to take it more. So I mean the it should not be end before the examination, prior to the examination. So please keep in mind that and then take a subscription. Okay, so guys, thank you very much, and let's start with our lecture. Our topic, our today's topic is a plant growth. uh, I don't know, RR. So plant growth means, guys, it doesn't mean that how actually plant grow from the lower length to the higher length. Okay, plant growth is started from the development of embryo. Okay, so development of embryo means, if you see, when the sperm and ova comes together. Okay, we know that the sperm and ova, they comes together. For example, ova and the pollen. Okay, they comes together and they form embryo or zygote, you can say. Okay, zygote inside of a seed. So if you open the seed, Okay, for example, you just open a seed. So any seed, for example, this is a dicot. When you open a dicot, you will find something here. Let me change the color. So red color, let's see. So something here. Okay, so this is nothing but the embryo. Okay, this is nothing but the embryo. So guys, when you see the embryo is already there inside of a seed, that means your the fertilization process is completed. Now we are going to see how this embryo start growing to the plant like this. Okay, step by step. Okay, but the first step, very first step, which is not in this the picture is here. Actually the embryo which is coming out of the seed, and it is having few roots. Okay, few different roots. Few different roots are there, which are very necessary to understand, for the understanding the plant growth. Okay, so let's start with that root. So guys, what is seed? First of all, try to understand what is seed. Seed, you know that it is somewhere found inside of the fruit in angiosperm, and it is open in a gymnosperm. Okay, but what is seed? Seed is formed by the process of reproduction. Okay, it is formed after the fertilization. Two gametes comes together, one yon, second yon comes together, and they form a seed. It is product of uh, ripened ovules. Ripened ovules means when the fertilization occur on the flower. Okay, fertilization occur on the flower. So there is a female part on a flower. We know that looks like this flower part. Okay, so this flower part shape part is a female part which is present on a flower. Okay, so flower is like this here. So here is a ovule present inside of the ovary. Okay, so this ovary convert itself into the fruit. It convert itself into the fruit. So ovary पासून काय तयार होणार मित्रांनो? फ्रूट. आणि ओव्ह्यूल पासून काय तयार होणार आहे? सीड. Okay, so seed is a product of ripened ovules. Okay, embryo is developed from the zygote, and the seed coat is from the integument. Okay, जे integument होते fertilization process मध्ये तुम्हाला सापडेल. तुम्ही जर लेक्चर शोधलं तर तुम्हाला fertilization process मध्ये integument सापडतील. नसेल तर आपण आणखी एक दोन ठिकाणी पाहणार आहोत. Okay, so this integument convert itself into the seed coat, and zygote is formed from the embryo. So embryo finally convert into the zygote finally convert itself into the embryo, and integument convert itself into the seed coat. Okay, seed is an embryonic plant. Seed is also called as embryonic plant. Embryonic plant means just develop व्हायला सुरुवात झाली किंवा काही सेल्स पासून तयार झाले की ज्याच्यामध्ये डिफरन्शिएशन झालेलं नाही. Okay, it is from 12th standard guys. Small seed is germinating into the seedling and grows into the huge tree. Okay, so when seed is a small, it is germinate. Germinate means, let's say for example, this is seed. Okay, when it germinate, it looks like this. This is soil layer, root, and shoot. This is called as a germination. Okay, germination required few type of the favorable conditions, and finally it grows into the huge trees. Okay, so let's see. Yeah, small birds, but birds. This word is not good to use here, beta. Okay, so that small bird and the bird like things we will see where we can use that bird and other words. Okay, that is not good word to use here. It will definitely reduce your mark if you use that bird like a thing here. Okay, so try to understand the meaning of words and the very important thing, the definition of a word. Okay, each and every word is having its own definition. If you use any word anywhere, so it will reduce your mark when you are attempting the theory examination. Okay, but when you are attempting any MCQs like examination, so may it increase your confusion. Okay, so let's see guys. सीट डॉर्मन्सी. सो फर्स्ट ऑफ ऑल व्हॉट इज मीन बाय डॉर्मन्सी. ओके, आपण घरामध्ये सीड ठेवतो, म्हणजे आपण ज्वारी बाजरी वगैरे विकत आणतो, सीडच आहे. ठीक आहे, तांदूळ वगैरे सीडच आहेत सगळे, शेंगदाणे असतात, वेगवेगळ्या डाळी किंवा डाळीच्या आधी जर तुम्ही पाहिलं हरभरे किंवा मूग मटकी घरामध्ये ठेवतो, सीडच आहेत सगळे. मग हे सगळे सीड आहेत, तर त्यांच्यापासून लगेच प्लांट का तयार होत नाही, लगेच ग्रो का होत नाहीत ते. ओके, घरामध्ये जर तुम्ही पाहिलं तर ह्युमिडिटी वगैरे सगळं मेंटेन आहे, मग का ते स्वतःला कन्वर्ट करत नाहीत प्लांट मध्ये. ओके, सो दॅट इज द ऍक्च्युअली वी कॉल इट अ डॉर्मन्सी. So let's see what is actually the dormancy. There is slightly difference what I am talking and the dormancy. There is a very slightly difference, but you can compare. Inability of a viable seed to germinate into the suitable environmental condition is known as a seed dormancy. Okay, so seed dormancy when seed is a viable. Okay, seed व्यवस्थित आहे, सगळं व्यवस्थित आहे, सुटेबल एन्व्हायरमेंट सुद्धा दिलाय, तरी सुद्धा जर जर्मिनेट होत नसेल तर त्याला आपण डॉर्मन्सी म्हणतो. डॉर्मन्सी म्हणजे इनॅबिलिटी ऑफ जर्मिनेशन. ओके, when in a suitable environment, if environment is suitable, seed is viable, still it is not going to germinate. Means, for example, this is a seed, you kept it into the soil. Okay, water is there, humidity, oxygen, carbon dioxide, everything is there, temperature is there, still it is not going to germinate into the plant. Okay, but seed is still looks like this after a few days, so it is called as a dormancy. Why? It is inability of a seed to germinate into the suitable environmental condition also. Okay, so growth and development of embryo is arrested temporarily during dormancy. In a dormancy, if you see, it is a phase of a life of a seed. It is a phase where development, growth and development of embryo is arrested. Arrest करता ना रे, is arrested. Okay, so your embryo is not going to germinate now because of the arrested growth. Okay, because of arrested growth, your embryo is not going to germinate, and this is called as a seed dormancy. Okay, why it is unnecessary? Let's see. And the first of all, what is the cause of a seed dormancy? The first cause is seed coat. When seed coat is more hard, it is difficult to come. It is difficult for embryo to come out of the seed coat. Okay, सीड कोट जर हार्ड असेल ना, तर त्यामधून एम्ब्रियो आतून बाहेर येत नाही, अडकून बसतो आतमध्ये. तर ते पहिलं रिझन आहे मित्रांनो. दुसरं आहे प्रेझेन्स ऑफ इमॅच्युअर एम्ब्रियो. समटाइम एम्ब्रियो इज नॉट कम्प्लिटली मॅच्युअर अँड it is not able to germinate itself into the plant. So the second reason. And the third, germinating inhibitors. Let's see, germination is a process, guys. Okay, germination, it is a process. It is controlled by the different type of chemicals and hormones. It may be inhibited. Okay, so this germination process may be inhibited, and if inhibitors are present, it will definitely block the process of germination. Okay, but if you say this type of the causes can be overcome, it can be a break. Okay, so the breaking of dormancy is a possible. How? Let's see, the few things are there. The first thing, breaking of dormancy, the first thing here, हार्ड सीड कोट. ओके, मग यासाठी काय केलं जातं, सीड कोटला ट्रीटमेंट दिली जाते. कधी कधी हॉट वॉटर ट्रीटमेंट असते, कधी कधी त्याच्यामध्ये थोडंफार किंवा आपण त्याला काय करतो, रब करतो. ओके, रबिंग, rubbing of seeds, presence of immature embryo, we can't do anything. Okay. Germinating inhibitor, so washing of seed prior to the planting. Okay. सीड प्लांट करायच्या आधी एकदा काय करायचे, व्यवस्थित वॉश करून घ्यायचे. सो द केमिकल विच इज प्रेझेंट ऑन द सीड कोट, which is sometime it is acting as an inhibitor, so it will remove from the seed coat, and now your seed is ready to germinate. Okay, so it can be a overcome dormancy, can be a break. Okay, and there are few hormones also which can used as a dormancy breaker. So when we start the study of hormones of a plant, so we will definitely discuss which hormones and how we can use uh, to uh, to break the dormancy of seed. Okay, let's see the seed germination. What is the germination actually? In a process of a seed germination, a resuming of metabolic activity in a dormant embryo and the growth to be a seedling. Okay, so when two things comes together, yeah beta, RR, if you want to become a doctor, uh, you want to learn about physics and chemistry also, my dear friend. Okay, so a plant is a part of a biology. Physics and chemistry, these are the different subjects, still you have to learn that. So it doesn't matter you are going to the doctor of a human or the animal, so it is necessary to understand few process. Okay, so there is design, your syllabus is already designed, and it is very, I mean the it's like a steel frame. Okay, you can't crack it, guys, and your syllabus is a beautifully planned, guys. So the having this type of a question in your mind is not good for your dreams. Okay, it is not good for your the dream of a doctor. It doesn't matter what you want to become, guys, but the syllabus which is coming in front of you, or the syllabus which is already in front of you, is the final for you. That is actually the first thing you have to do. So first of all, complete it, and then become whatever you want. Okay, otherwise it is difficult for you. If you say that I want to become a doctor, so I'm just going to be I'm just going to study physiology of a human, so this will not work, definitely. So you can't become a doctor if you are not a, student of a NEET is compulsory for a doctor. Okay, so let's see, guys. In a process of resuming metabolism, so when the ova and the sperm means pollen here. Okay, about the plant we use a term pollen. They comes together, and the embryo is a form, zygote is a form. Okay, so this zygote is formed. That time the metabolic activity, if you see metabolism, that was running very fast, but the plant, before the germination and after the zygote, it stopped the metabolism. Okay, so metabolism is not working now. When you put this seed, after the formation of a seed, metabolism is completely stopped. When you put this seed into the soil, goodbye beta. So this is seed, you are keeping it in a soil, so now it start germinated. Okay, so what is meant by germination? So here your seed is just become a metabolically active. First thing, become metabolically active. Okay, first step in a plant growth, the first step in a plant growth is the activation of metabolism, but to activate the metabolism in a seed, the very initial, very important stage is absorption of water, okay, by imbibition and the swelling process, okay, sorry, by imbibition, and then seed become the larger by a swelling process. Okay, so guys, what is meant by imbibition? Imbibition means absorption of water by the hydrophilic molecules, like cellulose is hydrophilic. Your the component which is present in a cell wall of a plant is a hydrophilic component. Most of them are hydrophilic. So imbibition, by the imbibition, if you put a seed into the water, automatically start absorbing the water, and it's called as a rehydration. After the rehydration, I mean the absorption of water, seed automatically resume its activity. Okay, resume its activity of metabolism. So first thing is absorption of water. Second is resuming of metabolic activity. What is meant by metabolic activity? Metabolic activity means activation of few enzymes, like hydrolytic enzymes which become active. So hydrolytic enzymes, why it is needed, guys? Let's see. Okay, what is meant by hydrolytic first of all, and the second thing, which enzymes? So we know seed, if you see seed is good for health. Okay, why you eat a seed? So it is because of the high level of protein is stored inside of a seed. High amount of lipid is there, high amount of carbohydrate is there. So you prefer seed in your meal. Okay, so not necessary each and every seed is having a very high amount of protein, lipid and carbohydrate, but depending upon the seed, this value is a change. Sometimes few seeds having more protein as compared to the lipid and carbohydrate, some having more carbohydrate as compared to the protein and lipid, depending upon the plant, depending upon the seed. Okay, so let's see guys, these hydrolytic enzymes like proteases, first enzyme for example, proteases. So proteases act on a protein and they start the digestion of a protein so that they can form energy, they can synthesize the energy. Second, lipases. So here they start digestion of a lipid, so they can use it as a source of energy. Third, amylases type enzymes, they start the digestion of a carbohydrate, so we will have, I mean the seed will have the glucose, and which result into the pyruvic acid, or you can say the energy after the glycolysis and other process energy. Okay, so metabolism of stored food, that is the metabolism of stored food until the activation of photosynthesis. So this food is required for the germination till activation of photosynthesis. ओके, तुम्हाला माहिती आहे जर तुम्ही जर्मिनेटिंग सीड पाहिलं, जर्मिनेटिंग प्लांट पाहिलं, तर त्याच्यामध्ये तुमच्या लक्षात येईल की ते प्लांट लगेच सीड मधनं, फॉर एक्झाम्पल हे सीड आहे आणि याच्यामधनं जे प्लांट बाहेर येतं तर ते लगेच ग्रीन नसतं मित्रांनो, बरोबर. ग्रीन नसेल तर त्याला लाईटचा काही फायदा नाही, फोटोसिंथेसिस करता येत नाही. जोपर्यंत फोटोसिंथेसिस ऍक्टिव्हेट होत नाही तोपर्यंत त्याला या सीड मधनं मटेरियल प्रोव्हाइड करावं लागतं, आणि म्हणून सीड मध्ये एक्स्ट्रा मटेरियल भरपूर असतं की जोपर्यंत प्लांट ग्रीन होत नाही तोपर्यंत त्याला या सगळ्या गोष्टी अवेलेबल असल्या पाहिजे. आणि म्हणून सीड टाइप ज्या गोष्टी आहेत तर त्या आपण प्रेफर करतो मिल मध्ये, कारण त्याच्यामध्ये भरपूर सगळ्या गोष्टी आहेत की ज्या तुमचे बायोमॉलिक्युल्स आहेत त्याच्यामध्ये स्टोअर्ड फॉर्म मध्ये असतात, मग तुम्ही खाता सीड्स. ओके, त्यामुळे बऱ्याचशा गोष्टी, सगळ्या डाळी वगैरे ज्या आहेत ना घरामध्ये आपण खातो त्या सगळ्या सीड्स आहेत मित्रांनो. ठीक आहे, सीड्स खाणं आपल्याला आपण जास्त प्रेफर करतो. So let's see the type of germination. Okay, when you plant a seed, so there is few style related with the germination. I mean when the embryo is developed from the seed into the plant, so it is also having its style of development. Okay, so the first style of development is hypogeal germination. Hypo means below, and geal or geo means soil. Okay, so it germinate below the soil. How? The epicotyle elongate quickly, pulling the plumule out of the soil. Plumule means, let's see for example this is a seed. Okay, So it develop like this. So one uh, a growth is coming out of the soil, means moving toward the sky, and this is moving toward the soil, means the root. Okay, so this root forming part is called as a radical. Okay, and the shoot forming part is called as a plumule. Okay, so plumule comes out of the soil, so you can see plumule is coming out of the soil here. Cotyledon do not come, cotyledon still here, as a seed it is still inside of the soil. Cotyledon do not come over the soil surface. Okay, so this is start. I'm going to make a start on this process, because in a second type of, or second style of germination, we will have the cotyledon out of the soil. Okay, so most of the monocot germinate by this style, most of the monocots, and few dicot like a mango and the groundnut, pea. Okay, so here is comma, I just forget to give pea, mango and groundnut. pea means वाटाणा, ठीक आहे, आणि ग्राउंडनट म्हणजे शेंगदाणे. तर हे जे प्लांट्स आहेत, तर हे या स्टाईलने ग्रो होतात, आणि ही स्टाईल आहे प्लांटची ग्रोथची. ठीक आहे, असं यात लगेच अवघड समजण्यासारखं काही नाहीये, स्टाईल आहे फक्त. कोणत्या कोणत्या स्टाईलने प्लांट ग्रो होतात, ज्या सीड पासून प्लांट तयार होताना काय कसे कसे प्रकार आहेत, आणि त्याच्यामध्ये काय काय पद्धती आहेत. तर त्यातली ही पहिली स्टाईल आहे हायपोजियल जर्मिनेशन, वेअर the seed and the cotyledon remain under the soil, they are not coming out, but the plumule is coming out of the soil, and the both side of a plumule are still there inside of the soil. Okay, so you can see the plumule is moving like this. This is a plumule which is forming a first true leaf, and this is your cotyledon, part of a cotyledon which is coming out, and this is not this cotyledon part is not permanent, it is temporary. After the few days or the small time, it automatically remove from the plant, it automatically come out of the plant. Okay, so the second type of germination, or you can say the second style of germination, is a epigl. Ep means above and gl means soils. So in the last style, if you say this cotyledon was inside of the plant, inside of a soil, which is not coming out of the soil, but here you can see it is coming out of the soil. Okay, so when, if you say the cotyledon and the radical, the portion between the cotyledon and radical is called as a hypocotyle. Okay, so here is a hypocotyle, and the portion which is coming out from the cotyledon and moving to the next, I mean the here you can see after the cotyledon, above the cotyledon, the portion is called as epicotyle. Okay, so epicotyle and hypocotyle, try to understand. Hypocotyle is below the cotyledon, that's why hypo, and ep means present after the cotyledon, above the cotyledon, so called as epicotyle. Okay, काय रे हे ओके, हे काय मित्रांनो, तर यामध्ये लक्षात घ्या की प्लांटचे फक्त दोन पार्ट आहेत, हायपोकोटाईल आणि एपिकोटाईल. कन्फ्यूज व्हायचं काही कारण नाही, छोटी गोष्ट आहे. जे सॉइल पासून वरती आहे and below the cotyledon, above the soil and below the cotyledon is called as hypocotyle, and after the cotyledon, I mean the above the cotyledon, the part is called as epicotyle. Okay, so here what happen, hypocotyle elongate quickly pulling the cotyledon and plumule above the surface. Hypocotyle elongate. So where is hypocotyle? You can see here. This hypocotyle elongate, and which pulls cotyledon and plumule both out of the soil. Okay, plumule and cotyledon both out of the soil. Cotyledon become green and start photosynthesis. So your cotyledons become a green and start photosynthesis. This photosynthesis cotyledon are looks like a leaf, and this is called as a foliage leaf. These are not permanent, these are the temporary leaf, and it is called as foliage leaf. लक्षात ठेवायचे, foliage leaf तुम्हाला पेपरला विचारलं जातं. What is foliage leaf? It is nothing but the temporary leaf formed from the cotyledon. It is formed from the cotyledon or the modification of a cotyledon. Cotyledon थोड्या वेळासाठी कॉटिलेडॉन थोड्या वेळासाठी फोटोसिंथेसिस करतात जोपर्यंत ट्रू लीव्स तयार होत नाहीत. ही ट्रू लीव्स तयार होत नाही. ओके, मेनी मोनोकॉट्स लाईक अ कॅस्टर, टॅमरिन एक्सेट्रा प्लांट्स आर growing by this style of EPGL germination. Okay, so this is second style of germination. Here is actually the key in this germination, and this is the second, you can say, you can write it also in your examination. Okay, so let's see type of germination. Third type, third fashion or third style of germination is viviparous. Okay, so this is a very rare process, guys, and it is only followed by the plants which is halophytes, or you can say the mangroves plants. Okay, mangroves म्हणजे दलदलीच्या प्रदेशामध्ये जे राहतात की खाली खूप चिखल असतो. सीड जर वरतून पडलं तर चिखलामध्ये खाली बुडून जातं ते. त्याच्यावरती एकदा चिखल तयार झालं की त्यातून प्लांट तयार होणं अवघड होतं. मग अशा पॉइंटला काय केलं जातं की प्लांट डायरेक्ट सीड वरतीच तयार होतं. जोपर्यंत सीड प्लांटला आहे तोपर्यंतच प्लांट तयार होतो. त्याला विविपॅरस म्हणतो आपण. विविपॅरस इज अ स्पेशल टाइप ऑफ जर्मिनेशन यू कॅन सी ओनली इन अ mangroves plant or the halophytes. Okay, grow in a salty and marshy soils. Mangroves and halophytes म्हणजे grow the plant which is growing in a salt and marshy soil. Marsh म्हणजे दलदलीच प्रदेश. ओके, सीड जर्मिनेट इनसाइड. दलदल म्हणजे खाली खूप चिखल असतो. त्याच्यावरती जर सीड किंवा फ्रूट पडलं, ओके, तर काय होतं मित्रांनो, त्याच्यावरती सीड पडलं की सीडला ना सीड किंवा फ्रूट दोन्ही पण आहे ना खाली चिखलामध्ये जाऊन बसतात. ठीक आहे, आणि एकदा दोन्ही चिखलामध्ये जाऊन बसले की त्याच्यामधून सीडला जर्मिनेट होऊन प्लांटला बाहेर यायला अवघड जातं. त्यामुळे अशा कंडिशन मध्ये तुम्हाला या टाइपची एक अरेंजमेंट म्हणा किंवा मॉडिफिकेशन म्हणा प्लांट मधल्या जर्मिनेशनचं, तर या टाइपची जर्मिनेशन पाहायला भेटेल. नाहीतर हे जर्मिनेशन मोस्टली जे सॉइल प्लांट आहे जे तुम्ही आम्ही पाहतो त्याच्यामध्ये नाहीये. फक्त मॅनग्रोव्ज वाल्या प्लांट्स मध्ये थोडाफार आपल्याला हा प्रकार पाहायला भेटतो. लिमिटेड प्लांट आहेत, त्यांचे एक्झाम्पल मी देतोय खाली. सीड जर्मिनेट इनसाइड ऑफ द फ्रूट. हे बघा प्लांट मध्ये हा फ्रूट वरच आहे. तुम्ही जर डायग्राम मध्ये पाहिलं तर फ्रूट आणखी प्लांटलाच लटकलेलं आहे. इथं आणखी प्लांट सोबत पॅच आहे. ओके, seed germinate inside the fruit still attached to the plant. Okay, so fruit is attached to the plant and seed is start germinating there also. Okay, so elongation hypocotyle result into the growth of a radical. Hypocotyle grow व्हायला सुरुवात होतो आणि radical ची growth होते. Radical come out of the fruit. Okay, so the radical which is coming out of the fruit, bulky weight of a seedling detached from the parental plant. आणि याचं वेट वाढत चालतं. तुमच्या लक्षात येतं की हे वेट किती होणार आहे हळूहळू. हे रॅडिकल खाली आलं, याचं वेट एवढं वाढतं की हा पार्ट इथून कट होतो. म्हणजे जे फ्रूट आहे तर ते प्लांट पासून काय होणार आहे मित्रांनो, डिटॅच होणार आहे. आणि ते प्लांट पासून डिटॅच झालं की सीडलिंग फॉल्स व्हर्टिकली डाऊनवर्ड. व्हर्टिकली डाऊनवर्ड म्हणजे नेहमी ज्यावेळेस पण मातीत पडेल, फॉर एक्झाम्पल खाली चिखल आहे असं, ज्यावेळेस पण हे मातीत पडेल ना त्यावेळेस ते असं पडतं. प्लांट फक्त याला ग्रो करून बल्की करत नाही मित्रांनो, त्याचा सेंटर ऑफ ग्रॅव्हिटी सुद्धा मेंटेन केला जातो. आणि इथून लगेच तुम्हाला एक सीड जर्मिनेट होऊन, किंवा तुम्ही म्हणू शकता सीड तर आधीच जर्मिनेट झालेलं आहे. ओके, हे प्लांट ग्रो व्हायला सुरुवात होते. हे यासाठी आहे की यानं खूप खोल जाऊ नये खाली. जर खाली खूप खोल गेलं फ्रूट किंवा सीड तर इथून प्लांटला बाहेर येईपर्यंत लिमिटेड आतमध्ये फूड असतं. आपल्याला माहिती आहे की फूड लिमिटेड आहे. मग इथून बाहेर येईपर्यंत फोटोसिंथेसिस करेपर्यंत जर फूड संपलं तर प्लांट जर्मिनेशनच्या प्रोबॅबिलिटी कमी होत चालते. आणि ही प्रोबॅबिलिटी मेंटेन ठेवण्यासाठी आपल्याला कशाची गरज आहे मित्रांनो, या टाइपच्या अरेंजमेंटची गरज आहे. सो दिस अरेंजमेंट इज फाउंड ओनली इन मॅनग्रोव्ज. इफ यू सी द एक्झाम्पल्स, rhizopora and Soneraceae are the example. Soneraceae and the rhizoporas are the example of viviparous germination. It is special type of germination you can't find in each and every plant, but you can find this special type of germination inside of the mangrove. Okay, let's see guys, what is a growth. Growth is a permanent increase in a size. Okay, so permanent increase, let's see the word This is very important word, permanent increase in a size, volume and the dry weight. Okay, so all things are growing here like a size, volume and the weight also. Okay, so the number of molecules and the matter which is inside of a cell is also growing. Change is a permanent and irreversible. When the growth is happen, so it is irreversible. Okay, so it can not move in a opposite direction. It is irreversible and the permanent. Growth is intrinsic, that is the innate, which is coming from the internal activity, or you can say it is a result of a metabolism. Okay, so the sentence means like that. It is a result of a metabolism. It is an intrinsic process caused by the internal activity like a metabolisms. Okay, the phases, if you see that there are three phases, three main phases of growth: 1, 2 and 3. First is a formative phase, second is the elongation phase, and third is a maturation phase. Okay, so let's see these three phases in 10 to 15 minutes, and we will going to stop. Okay, first is a formative phase. So it is actually the phase of a cell division. Phase of cell division. Okay, so in a cell division, if you say the cells are cell divides and the number of cell increases. Okay, so in a cell division, this is a process of a cell division. It is called as a mitosis. Okay, why mitosis? Because both cells are having 2n. Okay, which is coming started, the first cell, parental cell is a 2n and the final cell is a 2n, so it is mitosis. Meristematic cells undergo the mitosis process. Meristematic cell, the meristematic the tissue which is actually the very active in a division. Okay, which is present on the both end of the plant. Okay, so root meristem is also and the shoot meristem is also there, which is responsible for the growth of a plant. Now cell form, sorry, new cell form are the thin walled and not showing vacuoles, prominent nucleus and the dense cytoplasm. So this is actually the property of meristematic tissues or the meristem cell. Meristem cells are specialized for the cell division. Okay, so guys, try to understand. Meristem cells are specialized for the cell division. So these cells do not have very strong cell wall for the uh, I mean if the strong cell wall is there, so it is difficult for a cell to divide it, and that's why the cell wall is a thin. Vacuole is not necessary because the it is a highly metabolically active cell. Okay, so it does not require, it does not, it don't want to store something inside of a cell, so it continuously the metabolism is very fast, and that's why the cell is always engaged with the cell division process. Okay, addition of a new cell slightly increase the size of a plant. Okay, so here if you see the process is started from a one cell and it results into the two cell. Okay, but sorry guys, after the cell division, if you see, number of cells are increased but the size is not increased too much. Why? Let's see, because reason is your second phase, elongation phase. Okay, so let's see what happened in a first phase. First phase, one cell divide into the two cell. Okay, so it is your formative phase or the first phase, cell division phase, but the size do not become more due to the, if you say the after division these two cells are not same size, not having the same size of a parental cell, but in the process, second process or the second stage, elongation phase, these cells undergo the enlargement. They are now absorbing water and the nutritions and the food from the other part of a cell, uh, sorry, other part of a plant. Enlargement is a more in a linear direction result in a elongation. So this enlargement is not like this, okay, but this enlargement is like this. This cell is also enlarged like this, okay. So if you see, cell are not going to uh, looks like a bulky, but they are looks like the cylindrical, okay. And that's why height of a plant is increases, height is increased. Cell enlargement takes place by the synthesis of a protoplasm. Protoplasm is a liquid inside of a cell, and the large quantity of a solute. Solute means the salt and the other thing like a nutrition, okay. These materials are moving inside of a cell, and that's why the osmotic pressure increase, and it results into the absorption of automatically absorption of water, okay. When salt concentration increase inside of a cell, cells start absorbing water from the surrounding, and that's why it become a turgid and the enlarge in a linear direction, okay. So solid concentration favor endoosmosis. That is what I just told you. When solid concentration increases, cell start absorbing water from the surrounding. Cell become turgid. Expansion of a cell by adding more cellulose in a wall. Okay. So when the cellulose is added in a wall, the cell start expand. Vacuole appear. So now this cell is after the second phase of elongation, cell is not going to enter into the again cell division process. So cell start making the organs like vacuoles and the cell wall, okay. It results, so this all process, all stages results into the considerable increase in size, weight, and as a whole plant, okay. So it results into the considerable increase, okay. In a first stage, the cells are double, cells become double, but still there is not considerable increase in a size, but in a elongation process the size definitely increased, okay. And the last one is a maturation. This is final phase of growth, okay. What is meant by maturation and differentiation? The meaning is uh, similar. Elongated cell, which is coming from the second stage, okay. So the first stage we know, the formation stage, where the cell division happen, cell division first stage, and number of cells are now 2, okay. So after the elongation, it is a second stage of growth. Cell become elongated, and now this elongated cell differentiate into the various type of tissue by the process of differentiation, okay. It undergo the process of differentiation, and it result into the tissue. For example, this cell uh, become a parenchyma, collenchyma, or sometime this cell says I want to become a sieve tube of a vascular system, okay. So this cell convert itself into this special type of a cell. So specialization is the process of differentiation, okay. And this cell convert itself into this second special type. So whatever it is, for example, here is a scalenchyma and here is a parenchyma. These are the two different type of a cell. The one cell, I mean the same type of a cell become itself into the different type of a cell. So the attempting फिक्स और द स्पेशल फंक्शन इज कॉल्ड एज इलॉंगेशन. इलॉंगेशन म्हणजे कोणतं तरी एक फंक्शन अटेम्प्ट करायचं काही त्यात. मग फॉर एक्झाम्पल एखाद्या ग्रुप ऑफ सेल डिसाइड दॅट वी विल गोइंग टू बिकम अ झायलम, सो दॅट इज द स्पेशलायझेशन. झायलम म्हणजे एक सेल नाही, त्याच्यामध्ये पण 4 टाईप आहेत. मग त्याच्यामधले कोणता टाईप होणार आहे तो स्पेशलायझेशन झालं. प्रत्येक सेल आपलं आपलं स्पेशलायझेशन करून घेते, त्याला आपण डिफरन्शिएशन म्हणतो, ठीक आहे, किंवा मॅच्युरेशन. In this process, cell gain its permanent structure and function, okay. आपल्या बॉडी मध्ये सुद्धा हे डिफरन्शिएशन होतं. आपल्या बॉडीतल्या सेल डिसाइड करतात की फॉर एक्झाम्पल आय वॉन्ट टू बिकम द epithelial cell, I want to become a nerve cell. So whatever it is, at the time of a development, at the time of a embryo development, the cell find it's a lineage, or you can say that it's a root, or it's a differentiation process, and they start moving toward in that direction. Okay, so they finally get the permanent structure and the permanent function in an animal and in a plant also, but the plant is having one more chance after the permanent structure and the permanent function. If there is a requirement, cell can enter itself into the undifferentiated cell. ओके, म्हणजे प्लांट सेल परत अनडिफरेंशिएटेड मध्ये येऊ शकतात, ॲनिमल सेल येत नाहीत, म्हणून आपल्याकडे रिजनरेशन प्रॉपर्टी नाही, आणि प्लांट सेलला परत वापस येता येतं अनडिफरेंशिएटेड फॉर्म मध्ये, म्हणून प्लांटकडे रिजनरेशन प्रॉपर्टी आहे. तुम्ही जर प्लांट अर्ध्यातून तोडून टाकलं तर पूर्ण प्लांट परत येतं. तसं आपलं ह्युमन्सचं किंवा ॲनिमलचं नाही, हात पाय किंवा बोट वगैरे तुटलं तर परत येत नाही. त्याचं कारण असं आहे की डिफरेंशिएटेड झालेलं सेल परत एकदा डिफरेंशिएशन प्रोसेस मध्ये जात नाहीत, आणि मग त्याची जागा घ्यायला कोणी नसतं. त्यांना अनडिफरेंशिएट होता येत नाही, परत अनडिफरेंशिएटेड फॉर्म मध्ये जाता येत नाही. मग ते रिडिफरेंशिएशन आणि डिडिफरेंशिएशन दोन्ही प्रोसेस आपल्यात होत नाही, ह्या फक्त प्लांट्स मध्ये होतात, आणि म्हणून प्लांट कडे ही प्रॉपर्टी आहे. ठीक आहे. So sequence of development, let's see, this is I think the last slide. So meristematic cell, it convert itself into the stages like a differentiation, and we found the maturation cell. Okay, so in this the process of this is the complete process of differentiation. In this process you can see your stages of growth. The first stage here, cell division, that is the formation. Okay, cell division, expansion, maturation, and the mature cells by the senescence results into the death, and sometime due to the any reason or due to the any problem, this plasmatic growth or these growth cells become again meristematic by the cell division, and the cycle is started to increase the number of cells. Okay, आणि number of cells continue वाढत चालतात. तर या प्रोसेसला आपण डेव्हलपमेंट म्हणतो. ठीक आहे मित्रांनो. डिफरेंशिएशन आणि रिडिफरेंशिएशन, तर या स्लाइड्स मध्ये तुम्हाला काही गोष्टी लक्षात येतील की which is related to the plant and which is not present inside of the animal. Okay, so the first process, dedifferentiation. What is meant by dedifferentiation? Parenchymatous cells may regain the capacity of a divide, and the redivide is called as a differentiation. First of all, say the meristematic tissue was the first tissue which is having the capacity of a division. Okay, now it become a parenchymatous cell. For example, parenchyma, it become parenchyma by the process of differentiation. डिफरन्शिएशन म्हणजे काय, एक परमनंट शेप आणि परमनंट स्ट्रक्चर ऑक्युपाय केलं, पॅरेनकायमा मध्ये कन्वर्ट झालं. बट इफ देयर इज अ रिक्वायरमेंट ऑफ अ मेरिस्टेमॅटिक सेल, दिस पॅरेनकायमा convert itself into the meristematic tissue again, and this is called as a dedifferentiation. So dedifferentiation is a process reverse to the differentiation. Dedifferentiation is a process which is reverse to the dedifferentiation, and when this dedifferentiated cell again convert itself into the parenchyma or the collenchyma or the sclerenchyma, whatever it is, so it is called as a redifferentiation. Okay, so the first is a differentiation. Meristematic tissue to the any other specialized tissue is called as a dedifferentiation. Any other specialized tissue to again meristematic tissue is called as a dedifferentiation. And the after dedifferentiation, when meristematic tissue again convert itself into the any other special type of tissue, is called as a redifferentiation, and it is happen only in a plants, guys, and not in an animal like us. आपल्यासारख्या animal मध्ये होत नाही. नंदिनी सर MST CET 2020 में जनरल कैटेगरी में COEP CS ब्रांच जनरल कैटेगरी, Nandini COEP engineering college है बेटा. So I don't know exactly. Okay, exact if you say the exact, I mean you are asking the exact the percentile or the exact rank, so I don't know. Okay, interfascicular cambium, and the quark cambium is a form from the de-differentiation process. De-differentiation process is necessary for the formation of cambiums like interfascicular cambium and the quark cambium. These cambiums are necessary for the secondary growth. The product of a dedifferentiated cell or the tissue which lose the ability to divide and called as a dedifferentiated cells or the tissue, and event is called as a redifferentiation. Okay, so I already explained this dedifferentiation and redifferentiation here in the form of the process. Okay, हाँ एग्जाम होगी ना बेटा, एग्जाम क्यों नहीं होगी? सीटी सेल ने डिक्लेयर किया है और एक नोटिफिकेशन आ जाएगा तो मैं वीडियो बना के अपलोड कर दूंगा आपके लिए कि कौन सा सिलेबस होगा, कब एग्जाम होगी, ठीक है। मैं वीडियो बना के आपको जल्द से जल्द भेज दूंगा बेटा, ठीक है। एग्जाम होगा आपका, थ्योरी भी होगा, ये भी होगा, ठीक है, दोनों भी एग्जाम होंगे। The product of a de-differentiation, sorry, we already explained this de-differentiation and re-differentiation. Now let's see last thing, growth phase. So actually the growth curve, this is not a growth phase but this is a growth curve, and a bacteria, when you are studying a bacteria, most of the time you can see this curve, but it is used for the any other, I mean the plant cell and the animal cell also. So where the in a first phase you can see the lag phase where the growth is a very, if you see the size is not going to increase very fast with respect to the time, but after that as compared to the time the size is increasing very fast, is called as exponential growth. So this is actually the preparation phase, we call it a lag phase, and after the attempting the particular height or the particular growth, so again cell uh, maintain its count and not going to move toward the very high numbers, is called as the stationary phase. If there is if you see in a stationary phase the growth of a cell or the division of a cell, or the, you can say the making of a toxic material is also there, which results into the stopping of a growth. Second thing, if this material is going to be an increase, it will result into the death phase. Okay, so this is a death phase where the cells are dying because of the making of a toxic material or the waste material or the accumulation of a waste material. If we are talking about the ग्रोथ ऑफ अ सेल इनसाइड ऑफ अ लॅब, ओके, जर लॅब मध्ये असेल तरच असं होतं, एन्व्हायरमेंट मध्ये वेस्ट मटेरियल बाहेर फेकलं जातं, ठीक आहे। लॅब मध्ये काय होतं, वेस्ट मटेरियल त्या टेस्ट ट्यूब मध्ये असेल तर टेस्ट ट्यूब मध्येच राहतं आणि मग सेल स्टार्ट करते आणि स्वतःला डेथ फेज मध्ये घेऊन जाते, ठीक आहे। So guys, this was all about the plant growth. Okay, so let's say this is Crack Maharashtra CET channel. If you do not have subscribed, first of all subscribe it and click on a bell icon, and when you are taking a subscription, do not forget to use this code for the 10% discount, and this channel will give you the MCQs practice in a pool format, and the second thing, notes. Okay, so thank you very much and see you tomorrow at 11 a.m. Bye bye. Have a good time, guys. And the last thing, last but not least, Happy New Year, guys. I just forget to wish you Happy New Year. Bye bye.